Good day, everyone. I am Adrian H. Kalalang, BS Ed 2B, major in Sciences. And now, I am going to discuss the prokaryotic and the eukaryotic cells. First, what is a prokaryotic cell? A prokaryotic cell is a type of cell that does not have a true nucleus or membrane-bound organelles. Organisms within the domain bacteria and archaea are based on the prokaryotic cell, while all other forms of life are eukaryotic. However, organisms with prokaryotic cell are very abundant and make up much of Earth biomass. Next, an overview to the prokaryotic cell. Organisms that have prokaryotic cell are unicellular. They are called prokaryotes. The first prokaryotic cell has several elements that allow it to function as a living organism. First, the prokaryotic the prokaryotes are covered in a cell membrane. This membrane allows them to create a specific environment within the cytosol that allows biochemical reactions to take place. Second, these cell house both loose DNA and ribosomes. Through ribosomes and organelles, they are not bound by a plasma membrane. Together, the DNA and ribosomes work to produce the proteins that the cell needs to gather nutrients, reproduce, and even defend themselves in the face of predators or environmental changes. Prokaryotic are much smaller than the smallest eukaryotic cells. In general, a prokaryotic cell is smaller because it has less DNA to create the proteins needed to make an ultra-efficient membrane. So, the cell reach a size where they are can no longer important the number of nutrients they need for the volume of cytosol they contain. This is known as surface area to volume ratio limit. However, bacteria are much larger than viruses because they are actively carrying out the biochemical reaction of life within the cells. Next is the prokaryotic diagram. Most prokaryotic cells have nucleoid region, a number of ribosomes, a plasma membrane, and the cell wall made of peptidoglycan. Next, the prokaryotic cell structure. Prokaryotic cells do not have a true nucleus that contains their genetic materials as eukaryotic cells do. Instead, prokaryotic cells have a nucleoid region, which is an irregular shaped region that contains the cell's DNA and is not surrounded by nuclear envelope. Some other parts of prokaryotic cells are similar to those in eukaryotic cells, such as a cell wall surrounding the cell, which is also found in plant cells, although it has a different composition. Like eukaryotic cells, prokaryotic cells have a cytoplasm, a gel-like substance that make up the filling of the cell, and a cytoskeleton that holds components of the cells in place. Both prokaryotic cells and eukaryotic cells have ribosomes, which are organelles that produce proteins and vacuoles. Small spaces in cells that store nutrients and help eliminate waste. Some prokaryotic cells have flagella, which are tail-like structure that enable the organism to move around. They may also have pili, small hair-like structure that help bacteria adhere to surface and can allow DNA to be transferred between two prokaryotic cells in a process known as conjugation. Another part that is found in some bacteria is the capsule. The capsule is a streaky layer of carbohydrates that helps the bacterium adhere to surface in the surroundings. A prokaryotic cell is much smaller than eukaryotic cell. Through it is small much larger than the virus. The size of prokaryotic cell versus eukaryotic cells and a virus. The next is the prokaryotic cells parts. Unlike eukaryotic cells, prokaryotic cells have no distinct organelles bound by membranes. Instead, the many reactions the cell conducts happen within the cytoplasm of the cells. In fact, there are two main components that are present within all prokaryotic cells. The first is the cell membrane. This is a layer of phospholipids molecules that separates the inside of the cell from the outside. While not present in all prokaryotes, many secrete a cell wall used to protect and house the cell in an extra layer of proteins and structural molecules. The second part found in the prokaryotic cells is DNA. DNA is a basic blueprint for all life and is found within all cells. In prokaryotes, the DNA often takes the form of a large circular genome. This can be compared to the organized chromosomes, which are typically found within eukaryotes. This large circle of DNA directs with proteins the cell creates and regulates the action of the cells. Other prokaryotic cells can have a large number of different parts, such as a cilia and flagella, to help them move around. While these structures are similar in function to those found within eukaryotes, they often have a different structure. This suggests that the two types of cell have undergone very different selection processes and have independently evolved the structure. The next one is the characteristics of the prokaryotic cells. All prokaryotic cells have a nucleoid region, DNA and RNA, and their genetic material ribosomes that make proteins and cytosol that contains a cytoskeleton that organize cellular materials. However, prokaryotic organisms are very diverse group of organisms and come in many different shapes and sizes. These changes in structure typically represent changes in function, and these many different organisms occupy very different niches. Prokaryotic cells are usually between zero point 0.1 to 5 micrometers in length. Eukaryotic cells are generally much larger, between 10 and 100 micrometers. Prokaryotic cells have a higher surface area to volume ratio because they are smaller, which makes them able to obtain a larger amount of nutrients via their plasma membrane. Prokaryotic cells come in many different shapes and sizes, depending on how they have evolved and the roles they fill in an ecosystem. Next one is the example of prokaryotic cells, the bacteria cells. Bacteria are single-celled microorganisms that are found nearly everywhere on Earth, and they are very diverse in their shapes and structure. There are about 5 by 1,030 bacteria living in many different ecosystems on Earth, including in our own bodies. In the human gut, bacteria outnumber human cells 10 by 1. The cell walls of some bacteria contains peptidoglycan, a molecule made of sugars and amino acids that gives the cell wall its structures and is thicker in some bacteria than others. Bacteria contains certain structures unique to them, as previous mentioned, such as the capsule, flagella, and pili. Most bacteria have just a chromosome that is circular, which can range from about 160,000 base pairs to 12,200,000 BP. They also contain plasmids, which are small circular pieces of DNA that replicate independently of the chromosome. Some bacteria can form endospore. These are taught dormant structure that the bacteria can reproduce themselves to under a starvation condition when not enough nutrients are available. They do not need nutrients and are resistant to extreme temperatures, UV rays, and chemicals. When environmental conditions become favorable again, the endorsed spore can reactivate. Next one is the how do prokaryotic cells divides. Prokaryotic cells divides through the process of binary fission. Unlike mitosis, this process does not involve the condensation of DNA or the duplication of organelles. Prokaryotic cells have only a small amount of DNA, which is not stored in complex chromosomes. Further, there are no organelles, so there is nothing to divide. When a prokaryotes grow to a large size, the process of binary fission takes place. The process duplicates the DNA, then separates each new strands of DNA into individual cells. The process is simpler than mitosis. This means the bacteria can reproduce much faster than most eukaryotic organisms. Okay, the next one is the eukaryotic cell. First, what is eukaryotic cell? A eukaryotic cell contains membrane-bound organelles such as nucleus, mitochondria, and an endoplasmic reticulum. Organisms based on the eukaryotic cells include protozoa, fungi, plants, and animals. These organisms are grouped into the biological domain Eukaryota. Eukaryotic cells are larger and more complex than prokaryotic cells found in domains archaea and bacteria. Next one, an overview to the eukaryotic cell. A eukaryotic cell is one of the two different types of cells. Organisms that are based on the eukaryotic cells are called eukaryotes, and including plants, animals, fungi, and protists. The only organisms that are not based on the eukaryotic cell are organisms based on a prokaryotic cell structure. Those organisms are found in the domains archaea and bacteria. There are several differences between eukaryotic cell and prokaryotic cells that can help you fully understand what makes a cell eukaryotic. Moving forward, the characteristics of the eukaryotic cell. Eukaryotic cell contains a variety of organelles which performs various functions within the cell. All the organelles and stabilized are given physical support through the cytoskeleton, which is also involved in sending signals from one part of the cells to another. In eukaryotic cells, the cytoskeleton is composed mainly of three types of filaments: microtubules, microfilaments, and intermediate filaments. The watery solution that surrounds all the organelles is the cell is called cytosol. The next one is the eukaryotic cell cycle. The cell cycle is the life of the cell. During this cycle, it grows and divides. Checkpoints exist between all stages, so the proteins can determine whether the cell is ready to begin the next phase of the cycle. The first is the quiescence, also known as senses or resting, is a phase in which the cell is not actively dividing. It is also known as gap zero or G0. This stage is considered the start of the cell cycle, although is one the cell can reach and then stop dividing indefinitely, which ends the cell cycle. Cell in the liver, stomach, kidney, and nervous system are all examples of cells that can reach the stage and remains in it for long periods of time. It can also occur when a cell's DNA is damaged. However, most cells do not go into G0 stage at all and can divide indefinitely throughout the of an organism. Next stage is the interphase. During interphase, the cell grows and takes its nutrients in preparation for division. Interphase takes up about 90% of the cell cycle. It consists of three parts: gap 1, synthesis, and gap 2. Gap 1 is also known as the growth phase. The cell gets larger and increases in its stock of proteins along the organelles, such as energy producing mitochondria. Synthesis is the phase in which DNA replicates. During synthesis, the chromosomes replicate so that each chromosome is made up of two sister chromatids. And the end of this phase, there is double the amount of DNA in the cell. Gap 2 is another growth phase. The cell becomes even larger in order to prepare for mitotic division, and the remaining organelles are duplicated. Next is the mitosis or M phase, is when the cell begins to organize its duplicated DNA for separation into two daughter cells. The chromosome separates so that one of each chromosome goes into each daughter cells. This results in the daughter cells having identical chromosomes to the parent cell. Mitosis itself is divided into prophase, metaphase, anaphase, and telophase. Each phase marks various points of DNA in separation process. Mitosis is then followed by a process called cytokinesis, during which the cell separates its nuclei and other organelles in preparation for division and then physically divides into two cells. Moving forward, the examples of eukaryotic cells. The first one is the plant cell. Plant cells have a retic cell wall, which are easily seen in dark lines when viewed under microscope. Plant cells form thick cell walls. Plant cells are unique among eukaryotic cells for several reasons. They have a reinforced, relatively thin cells wall made of cellulose that help maintain structural support in the plants. Each plant cells have a large vacuole in the center that allows it to maintain turgor pressure. Turgor pressure results from the water in the central vacuole pushing outward on the cell walls. Plant cells also contains organelles called chloroplasts, which contain the molecules chlorophyll. This important molecule is used in the process of photosynthesis, which is how plants make sugar by using the energy found in light. Next example is the fungal cell. Like plant cell, fungal cell is also have a cell wall, but their cell wall is made of chitin, the same substance found in insect exoskeleton. Some fungi have septa, which are holes that allows organelles and cytoplasm to pass between them. This makes the boundaries between different cells less clear. Most fungi live underground or in decaying organic matter where the mycelial network contains millions of interconnected cells. Fungal cells often have holes between cells and called septa that allows the cytoplasm to flow freely between cells. Fungal cells often share cytoplasm through septa. Moving forward, the next example of eukaryotic cell are the animal cell. Animal cells do not have cell walls. Instead, they have only a plasma membrane that locks up the cell walls allows animal cells to form many different shapes. This allows for the processes of phagosynthesis, cell eating, or pinocytosis, cell drinking, to occur. Animal cells differ from plant cells in that they do not have chloroplasts and have many smaller vacuoles instead of the large central vacuole. The next one is the protozoa. Protozoa are eukaryotic organism that consists of a single cell. They can move around, eat other small organisms, and digest food within vacuoles. Some protozoa have many cilia, which are small, movable hairs that allow them to swim around. Others use large flagella structures that look something like a large tail to swim throughout the water. Some proteins also have a thin layer called pellicles, which provide support to the cell membrane. Next one is, what is the difference between prokaryotic and the eukaryotic cells? The difference between the prokaryotic cell and the eukaryotic cell is simple. Eukaryotic cells have a nucleus surrounded by a nuclear membrane and other membrane-bound organelles that perform specific function in the cell. These membranes form the endomembrane system, which creates a series of specialized chambers within eukaryotic organisms that can complete a diverse range of tasks. By contrast, a prokaryotic cell only has a cellular membrane with no membranes extending on the inside of the cell. All of the reactions within the prokaryotic takes place within the cytosol of the cell, while this makes the cell slightly less efficient. Prokaryotic cells still have a remarkable reproductive capacity. A prokaryotic reproduce through binary fission, a process that simply splits duplication DNA into separate cells without any organelles or complex chromosomes to reproduce. Most prokaryotic cells can divide every 24 hours or even faster with adequate supply of food. While many prokaryotic cells have adapted to free living within the environment, others have adapted to live within the gut of other organisms. The commensal organisms survive by breaking down molecules inside the gut and allow the organism they are living to the ability to digest a wider variety of food. For example, the human gut contains 2 to 3 pounds of bacteria that have evolved to help us digest complex carbohydrates, proteins, and fats. Okay, that's all for today. Thank you for listening. Hoping that you understand it very well. Again, I am Adrian H. Kalalang, Bachelor of Secondary Education, major in Sciences in Bulacan State University. Thank you and God bless.